Hello everybody, my name is Iman. Welcome back to my YouTube channel. Today we're going to continue our discussion about structure and function of large biomolecules. We've already talked about carbohydrates and lipids so far. So today we want to move on to talking about proteins. Now, proteins include a diversity of structures that's going to result in a wide range of functions. Proteins actually account for more than 50% of the dry mass of most cells, and they're instrumental in almost everything organisms do. Some proteins speed up chemical reactions, others play a role in defense, some play a role in storage and transport, while others play a role in cellular communication and movement. So the functions and the roles that proteins play are many. Life would not be possible without proteins, and especially the fact that some proteins serve as enzymes. Life would not be possible without enzymes. Enzymatic proteins, they regulate metabolism by acting as catalysts, chemical agents that selectively will speed up chemical reactions without being consumed by the reaction. Now, because an enzyme can perform its function over and over again without being consumed, these molecules can be thought of as the workhorses that keep cells running by carrying out the processes of life. Now, humans have tens of thousands of different proteins, each with a specific structure and each with a specific functions. Proteins, in fact, are the most structurally sophisticated molecules known, and the area of research studying many different proteins and their secondary structure and how it plays into their functions and how different mutations affect that secondary structure and that function is a huge research field, and it consists of a huge pool of research in biology and biophysics. And so proteins, because they're so structurally sophisticated, are always being investigated for their diverse function, their diverse structure, whether it be their secondary structure, and each type of protein has a unique three-dimensional shape that's also sometimes investigated for new proteins that are discovered. Now here what you see, this is a protein called myoglobin. This is the first protein to have its 3D structure known using X-ray crystallography. It has oxygen attached to it, and it's a common protein found in the heart and skeletal muscles. And when you exercise, your muscles are going to use up available oxygen. Myoglobin has oxygen attached to it, and so it provides an extra oxygen for the muscles to keep at a high level of activity for a longer period of time. And so this myoglobin plays a huge role in our heart and skeletal muscles, and it was the first protein to have its 3D structure known. Now, diverse as proteins are, they're constructed from the same 20 amino acids. And we're going to get into the 20 amino acids, but again, I want to emphasize just how many functions, the variety of functions these proteins have. They can be enzymatic proteins that will either selectively accelerate chemical reactions. They can be defensive proteins that protect against disease. We can have storage proteins that store amino acids and other things. We can have transport proteins that take one molecule to other cells that transport substances from one part of the body to another, and there's so much more. All right, now diverse as, again, proteins are, the building blocks of proteins are the same set of 20 amino acids that can be linked in many ways to form polymers. Now, the bond between amino acids are called peptide bonds. Now, let's see what the basic building block of proteins are. They're called amino acids. The typical structure of all amino acids is this. You have a carbon that's bonded to a hydrogen, to an amino group, and to a carboxyl group. We're just going to note it as COOH, and then to a side chain. This side chain is going to be unique. This is what distinguishes the 20 amino acids from each other. Other than that, they're all going to have the center carbon, this amino group, this hydrogen, and this carboxyl group. The only thing that's going to differ between each amino acid is this residue group. All right. So here is that amino acid structure. All right, all amino acids share this structure, and this side chain is what can be a variety of things for different amino acids. All right, so let's look at all 20 amino acids. Now, depending on your class, you might be asked to memorize these amino acids, and sometimes you won't be asked to memorize these amino acids. If you are pre-med, you definitely need to know the structure, name, the 3-letter code, and the 1-letter code that for each amino acid. But depending on your class for tests, that's up to your instructor. I just think it's pretty good to familiarize yourself as best as possible with these amino acids, especially if you are a STEM major. So, all amino acids share that common structure we talked about. Their side chains differ, and because their side chains differ, you're going to have some amino acids that are polar, some that are non-polar, some that are hydrophilic, some that are hydrophobic, some that have a positive charge, some that have a negative charge, some that have rings, and some that do not. All right, and so here we can start with a category. This category is amino acids with electrically charged side chains. All right, here are your positively charged side chains and your negatively charged side chains. All right, so here you have arginine, that's your residue. Here we have histidine. You notice that we have this ring for histidine. Here is lysine. All right, and for each of these, you have the name written for them, and you have the 3-letter code and also the 1-letter code for that amino acid. All right, for negatively charged side chains, we have aspartic acid and glutamic acid. All right, these have carboxylate groups that have negative charges, but they have varying carbon length chains between that lead up to that carboxylate group. All right, we have amino acids with polar uncharged side chains. These are serine, 309, asparagine, and glutamine. We also have special cases here. We have cysteine, glycine, and proline. Proline here is the only standard amino acid where the protein backbone is going to interact. The residue group is going to interact with the protein backbone twice, and it's going to form this ring in the protein or peptide structure. Then you have glycine here. Glycine is simply just a hydrogen right there. All right, it's the simplest amino acid. And then you have cysteine here. This is your only amino acid with a SH group. All right, you have one more other amino acid that has a sulfur group as well, and that's methionine right here. All right, and then you have your amino acids with your hydrophobic chains. All right, alanine, valine, isoleucine, and so on and so forth. All right, now the physical and chemical properties of the side chains determine the unique characteristics of a particular amino acid, and that's going to affect its functional role in a polypeptide. Now, the amino acids, again, are grouped according to the properties of their side chains. One group consists of amino acids with non-polar side chains, other ones with polar side chains, some with hydrophobic, others with hydrophilic. Another group can have rings and others do not. Some are acidic amino acids, others are basic amino acids, and so on and so forth. Now, with us covering these categories and some of the amino acids, what we can move into is how 2 amino acids can be added together. When 2 amino acids are positioned so that their carboxyl group of one amino acid is adjacent to the amino group of the other, they can then be joined together by a dehydration reaction. Notice how our dehydration reaction is the fundamental chemical reaction that seems to bind monomers of varying groups to form polymers. All right, we saw that with carbohydrates. We're seeing this with proteins now. All right, so we can have a dehydration reaction now with the removal of a water group. Right, so we can have a hydroxyl group from one and a hydrogen from another, and through a dehydration reaction, we can form a bond between one amino acid to the other. So with the removal of water, you have now joined two amino acids. The resulting covalent bond, this resulting covalent bond that's going to be between the carbon of what was once your carboxyl group and the nitrogen of what once was your amino group in your other amino acid, this is called a peptide bond. All right, and if you repeat this process over and over again, this process is going to yield a polypeptide, a polymer of many amino acids linked by these peptide bonds. So you can join two amino acids. If you have one carboxyl group aligned with one amino group, through a dehydration reaction, you can lose a water. And what you form there now is a peptide bond that links these two amino acids. You can repeat over and over again and form many amino acids together to form a polypeptide. Now, when a cell synthesizes a polypeptide, the chain that you form, all right, many things can happen to this polypeptide chain. All right. This polypeptide chain can fold spontaneously and assume the functional structure for that of a protein. Now, this folding is driven and reinforced by the formation of various bonds between parts of the chain, all right, which depend on what your sequence is for your polypeptides. All right, many proteins are we're ultimately going to build up to this protein structure, and many proteins have different kinds of structure that can be, spherical or long fibers. But it all starts with how your polypeptide chain can fold, and that is based off of what amino acid chain it can form. All right, so we're going to get into that now. There are over 20,000 different proteins in your body, and a protein specific structure determines exactly how it works. All right, in almost every case, the function of a protein is going to depend on its ability to recognize and bind to other molecules. Then we can ask, how do proteins have so much functional diversity based on only 20 amino acids? Well, the answer lies in the complexity of the primary, secondary, tertiary, and quaternary structures. So when you build a peptide chain, you have connected many amino acids to each other through a peptide bond. So just like how you would use alphabets, just how you would use the alphabet to build a word, right? You can take one letter, connect it to another, and now you've built a word, sat, for example. All right. You can build amino acids binded together to form a peptide or a polypeptide structure. All right, so this primary structure, it's only going to consist of amino acid sequences joined together to form a polypeptide. Even at this level, there is a substantial biochemical complexity that's included in this polypeptide chain. All right, this amino acids that are connected that form this, they can have different acidity, sizes, hydrophobicity, and charge at different points of this polypeptide. All right, and this difference in the amino acid sequence can result in different behaviors of your polypeptide. All right, so now you have this polypeptide sequence, all right, a chain of amino acids connected together. We can then move into the secondary structure. The secondary structure consists of the small scale 3D structures that's caused by the interaction between the amide groups of each of your amino acid that's linked together in your polypeptide. The major classes of interactions that can happen and result in the secondary structure are alpha helixes and beta pleated sheets. So what that means is that your polypeptide, all right, these amino acids that are linked together, can form some sort of secondary structure like an alpha helix, where they form this particular structure because there are some bonds, some interactions, some electrostatics that are happening between the groups of the different amino acids that form your polypeptide. And the two classes of this interactions that can happen within the polypeptide itself between the different amino groups are alpha helices and beta pleated sheets. All right, and the analogy to that is, well, you had letters before, and now you can form different words. All right, you can form different words from those letters that are going to be joined together in various ways. All right, so we went from letters in our primary structure to words in our secondary structure, in our analogy. All right, let me repeat. In our primary structure, you can think of this, all right, as the alphabet. You have many amino acids that are going to bind together to form long chains. Well, the amino acids of your chains, they're going to interact in certain unique ways based off of those amino acids, and they're going to form some sort of secondary structure because they're going to interact in unique ways. Just like your alphabet can come together to form words. All right, the main two classes of secondary structure are alpha helices and beta pleated sheets. Now, we can move into tertiary structure. Tertiary structure is the large-scale 3D conformation of the protein that's going to be caused by the numerous interactions between side chains of alpha helices and beta pleated sheets and even random coils. So this describes the manner in which the secondary structure elements can come together. Different secondary structures can come together and they're arranged in three dimensions to create a stable molecular entity. So these tertiary structures are primarily due to interactions between different secondary structures. All right, so notice how alpha helices can come together, interact with beta pleated sheets, interact with other alpha helices, and that forms our tertiary structure. So in our analogy, in the second structure, we formed words. In our tertiary structure, we bring together a bunch of secondary structure together to form our tertiary structure. What that means is we're bringing together a bunch of words to form sentences now. All right, and then last but not least, we have quaternary structure. Quaternary structure is the arrangement of several protein subunits in a complex. So if you can take different tertiary structures and interact them with each other and now form a quaternary structure. All right, so these are the 4 levels of structure for proteins. All right, let's move into nucleic acids. This is our last group of classes of large biomolecules that are found in all kinds of organisms. Nucleic acids store, transmit, and they help express hereditary information. Now, if the primary structure of polypeptides determines a protein shape, what determines the primary structure? Well, the amino acid sequence of a polypeptide is programmed by a discrete unit of inheritance known as genes. Genes encode proteins, and proteins dictate cellular function. Therefore, the thousands of genes that are expressed in a particular cell are going to determine what the cell can do because they encode for the proteins that and the proteins that your body needs to develop and build. All right, so genes encode for proteins, and we know that proteins dictate cellular functions. So we have these thousands of genes, and they're going to be that are expressed in a cell that are going to determine what the cell can do. Build the proteins we need. All right, build the proteins that our genes encode for so that our cells can function properly. All right. Now, genes consist of DNA, which belong to the class of compounds called nucleic acids. So what are genes? They consist of DNA that belong to a class of compounds called nucleic acids. Nucleic acids are polymers made of many monomer units called nucleotides. All right, so the building block of nucleic acids are nucleotides. All right, we're going to repeat that here shortly as well. The two types of nucleic acids that we're going to see are DNA and RNA. Deoxyribonucleic acid and ribonucleic acid. They enable living organisms to reproduce their complex components from one generation to another. All right, and unique among molecules, DNA provides directions for its own replication. DNA also directs RNA synthesis and through RNA controls protein synthesis. This entire process is actually called gene expression. All right, now we can say that DNA, RNA, DNA leads to RNA, like messenger RNA, which we're going to learn about, that are then used to build protein. So we have this kind of flow chart of DNA results in RNA, results in protein formation. Now, the building block of nucleic acids are nucleotides. Let's talk about DNA and RNA really quickly. DNA is the genetic material that organisms inherit from their parents. Each chromosome is going to contain one long DNA molecule. All right, and that's usually going to carry several hundreds or thousands of genes. When a cell reproduces itself by dividing, its DNA molecules are copied and then passed along from one generation of cells to the next. And so encoded in the structure of DNA is the information that programs all the cell's activities. All right, we're going to talk a lot about DNA later on, but essentially DNA is very important because the structure of the DNA contains the information that's going to program all the cell's activities. And you pass this down from a parent to a child, for example. RNA. How does RNA, the other type of nucleic acid, fit into gene expression? Well, RNA, the flow of genetic information from DNA to protein requires RNA. Each gene along a DNA molecule is going to direct the synthesis of a type of RNA called mRNA, or messenger RNA. And this mRNA molecule is going to interact with the cell's protein synthesizing machinery to direct the production of polypeptides, which are then going to fold into all or part of a protein. All right, and I just wanted to reiterate DNA and RNA and how they fit into our picture of building up to proteins. And so we can summarize the flow of genetic information as DNA to RNA to protein. All right, now that's a lot of information about DNA and RNA that you don't necessarily need to know at the moment. And more details specifically about nucleic acids and DNA and RNA and proteins are coming in later chapters. This is more of an introduction to the topic, but still, it's important to see how these things lead into one another. All right, so now we can move into our building block of nucleic acids, nucleotides. And nucleotide in general is composed of 3 parts. You have a 5-carbon sugar, all right, a 5-carbon sugar, a nitrogen-containing base, or a nitrogenous base that you see here, and 1 or more phosphate groups right here. So those are the 3 components that make up a nucleotide. That is the fundamental building block, or the monomer of nucleic acids. Now, the portion of the nucleotide that doesn't include the phosphate, if you just look at these 2 parts, excuse me, these 2 parts are also called nucleoside. Without the phosphate, they're called nucleoside. With the phosphate, they're called nucleotide. Now, there are 2 families of nitrogenous bases that we can have here. All right, we can have a pyrimidine. This is a group of nitrogenous bases that just have one 6-membered ring. They have one 6-membered ring that's of carbon and nitrogen atoms. All right, so here are are pyrimidines. Let's write that down. All right, this is a class of nitrogenous bases. They have 1 6-membered rings of carbon and nitrogen atoms, and you can just look at these 3 different pyrimidines. They're thiamine, cytosine, and uracil. Fantastic. The other class of nitrogenous bases are purines. Purines are larger, and they have a 6-membered ring that's fused to a 5-membered ring. All right, so these two right here are called purines. They're adenine and guanine. All right, now these specific pyridines and purines, they differ in their chemical groups attached to the ring. For example, you can notice the different groups that are attached to each of these rings. Even though they're purines and they have a 6-membered ring fused to a 5-membered ring, they have different groups attached to them. Now, adenine, guanine, and cytosine are found in both DNA and RNA. However, thymine is only found, let me do this in a different color. Thymine is only found in DNA, and uracil is only found in RNA. All right, now let's add the sugar to which nitrogenous base is attached. In DNA, all right, in DNA, the sugar is a deoxyribose sugar. In RNA, it is a ribose sugar. The only difference between these two sugars is that deoxyribose lacks an oxygen atom on the second carbon in the ring, hence the name deoxyribose. All right, that's our only difference between deoxyribose and ribose. This carbon right here in deoxyribose only has 2 hydrogens attached to it, whereas in ribose you have 1 hydrogen, 1 hydroxyl group. All right, in DNA we have deoxyribose, that's what the D stands for, deoxyribose nucleic acid, and in RNA we have ribonucleic acid. All right, so that's one difference between DNA and RNA. The building block, nevertheless, is a nucleotide, which has a phosphate group, a pentose sugar, and a nitrogenous base. You can have varying nitrogenous bases. You can have pyrimidines or purines. And the reason why you have a variety of this is when you build nucleic acids and when you build double-stranded, when you build up the double-stranded DNA, 2 different nucleic acid strands are going to interact with each other, and we're going to learn how these different nitrogenous bases are going to interact with each other to form, say, a double-stranded DNA. All right, so nucleotide has 3 parts: phosphate group, sugar, nitrogenous bases. All right, the sugar, you can have 2 different sugars, deoxyribose or ribose. This is what determines if you have DNA or RNA. And you also have a variety of nitrogenous bases. Fantastic. Now, let's talk about the linkage of nucleotides into polynucleotides. This is also going to involve, you guessed it, a dehydration reaction. And we're going to learn in detail about polynucleotides and DNA and RNA, because there's a lot of information that go into that we're not going to get into in this introduction chapter. But essentially, the linkage of nucleotides into a polynucleotide involve a dehydration reaction. All right, so to combine these monomers, these nucleotides together, we use a dehydration reaction. It's the same thing we saw with carbohydrates. To combine 2 monomers of carbohydrates, to combine 2 monosaccharides, you did a dehydration reaction. To combine 2 amino acids, 2 monomers of proteins, you use a dehydration reaction to form a bond between 2 amino acids. Here with nucleic acids, the monomers are your nucleotides, and to form a bond between your nucleotides, you also need a dehydration reaction. So in these polynucleotides, your adjacent nucleotides are going to be joined together by phosphodiester links, which consist of a phosphate group. All right, a phosphate group that links the sugars, to the sugars of your nucleotides. All right, so one nucleotide, all right, is bonded to another through this phosphate bond to the sugar. All right, that happens through a dehydration reaction. You lose water, and then between these two sugar and phosphate groups that are bonded together, all right, you form a bond. This is called a sugar phosphate bond when you continuously do this between many nucleotides. Now, the two free ends of the polymer, your nucleotide polymer, your polynucleotide, I'm sorry, are distinctly different from each other. This is one end of this polynucleotide. It has a phosphate group, and then the other end of your polynucleotide is a sugar. All right, one end has a phosphate attached to a carbon. The other end has a hydroxyl group attached to a carbon. All right, so you have this differing ends of your polynucleotide. One end has a phosphate attached to a 5-carbon. The other end has a hydroxyl group on a different carbon. So we refer to these ends as a 5-end and a 3-end respectively. And we're going to get into the importance of this when we discuss DNA. But the important thing that I want you to take away right now is that a polynucleotide has a built-in directionality along its sugar phosphate backbone. All right, somewhat like a one-way street. All along the sugar phosphate backbone are appendages consisting of these nitrogenous bases. This polynucleotide has a directionality, and that's going to be really important when we discuss DNA later down the line. All right. So essentially, these nucleotides you can form. You can build them together. You can build bonds between different nucleotides to get a polynucleotide. All right. And you can build DNA and RNA from that. RNA is usually a single-stranded sugar phosphate polynucleotide, whereas your DNA is a double-stranded sugar phosphate. And what you have here is one strand of polynucleotide that interacts with another strand of polynucleotide. And how do they do that? With the interaction between nitrogenous bases. All right, we talked about the different nitrogenous bases: adenine, guanine, thymine, cytosine, and uracil. Well, in DNA, adenine is going to form a bond with thymine. These are their complementary nitrogenous base pairings. This is the base pairing that happens. It happens between adenine and thymine. And what other base pairing that can happen is between cytosine and guanine. And that's how you get this double-stranded DNA, by the interactions and the formation of bonds between different nitrogenous bases. There's a lot more to come about DNA and RNA and all these other classes of biomolecules we've covered. This is simply an introduction to these 4 classes, their monomers, and how we can build up to polymers. Let me know if you have any questions. Other than that, good luck, happy studying, and have a beautiful, beautiful day.